several new that are reported for this. And what we can do now is we can ask cells, we can look at what is the immediate response to cells. It gains and loses different chromosomes. How is this impacting the abilities of cells to divide and go through the cell cycle? And look for, are there global or and karyotype specific cellular stress response? And also, what after you gain and lose a chromosome, what happened, which are selected for or against? And this, of course, is more than just a theoretical problem, because something like 90% of cancers are aneuploidy, gained or lost chromosome. We know that this is a critical part of the an irreversible part of the development of tumors. So again, nothing in this was pre-designed to be looking at how chromosomes were segregated, yet by thoughtful querying of the data, we were able to get at this question. And so I'll finish with what was really the biggest surprise, I think, in our data, which is we were looking at how did the cell respond as the mitochondria were perturbed? So mitochondria, remember, are these organelles that generate ATP. They're particularly remarkable because they were the consequence of an endosymbiotic event where bacteria got encompassed, basically archaebacteria encompassed alpha bacteria, and then that became dependent on the host for its survival and vice versa. And the thing was that when we perturbed this about 1,000 or so mitochondrial genes, when we perturbed the mitochondrial genes and looked at how the transcription in the nucleus changed, unlike that intricate structure that I got where we could break up the integrator, now basically there was a generic response. It wasn't able to distinguish. And that was, at first actually quite disappointing. But we noted that there were, in addition to the 1,000 or so mitochondrial coded genes that are expressed from the nuclear genome, their endosymbiotic history of the mitochondria meant that they actually have their own genome. They only express in humans 13 protein coding sequences and then some other tRNAs and ribosomal RNAs. So a small number of genes, but ones that are critical to the function of mitochondria. And now, if instead of clustering on the 1,000 or so nuclear coded mitochondrial genes, we just looked at how the transcription of these 13 genes went up and down, the level of these genes went up and down. Now we got this beautiful structure that when we looked at the function, again, very nicely separated the genes based on their function, on the function of mitochondria. And again, so here's a very nice example where we found all the genes that were involved in ATP synthesis, the machine that's directly is responsible for producing ATP in mitochondria, including finding a new and then unnamed component, TMEM242, which we went on, and others actually in parallel went on to show was an assembly factor for the ATPase. So why is this interesting? Stepping back, other than just sort of the discovery of these new components, because I think it tells you about the relationship between the mitochondria and its host. And the notion is that when you have specific defects in the mitochondria, this leads to a generic stress response in the nucleus that is not specific at all to the disruption. And then a stress-specific response in the mitochondria. So for example, loss of ATP synthase, loss of the large subunit, or loss of chaperones, each lead to distinct stereotypical response in the mitochondrial genome and this generic stress response in the nuclear genome. And taking one step further, what this has the potential for doing is allowing each mitochondria to respond locally to its own stress need. So it will both send out a generic stress signal to the nucleus that will affect all the mitochondria, and then a local response that will be specific to its own needs. So in that way, the cell can have 1,000 mitochondria or so, and be both monitoring the general health of the mitochondria, whereas each mitochondria can then be responding to its own stress. Now, people have studied the nuclear response to mitochondria. They've studied mitochondrial transcription for a long time. Yet it was really, this structure was really not apparent and had been essentially missed. I think it was large part, but yet when we looked at our data, it was really came out as a very strong and robust signal. And I think this is just a very nice example where in a data-driven way, you can get very strong signals that are hard to see through ad hoc types of experiments. So just more generally, we think that the approach of having rich phenotypes, transcription being one, but imaging or essentially anything that looks at the internal state of the cell in truly high dimensions with large-scale perturbations will increasingly be a tool for the principled discovery of the functional and cellular function. So with that, I'd really like to take a moment to thank the people who did the work, in particular, Joseph Rapogo, terrific MD PhD student, and then Ruben Saunders, a graduate student, drove much of these experiments, and Tom Norman, who was a postdoc in lab and now has his own lab at Memorial Sloan Kettering, actually worked with us closely on some of the data analysis approaches. In fact, I'll end and I'm going to let we'll see, it will be a small minor miracle if I'm able to switch.
Okay, yeah, as you can see, I'm Xiaojie, a postdoc on Bernardson's lab. So, actually, let me move this. So it's really my pleasure to present my work on mapping the field of single cells. So we actually have a preprint on this. And yeah, it's recently accepted. Sorry. Yeah, it's actually also recently accepted. So hopefully you will see the full version pretty soon. Of course, we also get a software package, the name. If you are interested, you can check it out. All right. So I think we are in a very exciting area for biology at this moment, thanks to the single cell genomics approach. Is able to generate to study the so example in this study generate of the chip, and then he uses the data set to analyze the cell type as well as the cell state transition. So although this kind of approach is very powerful, but it's very limited in the sense of it prohibits the discovery of the underlying regulatory mechanism. I always like to use this analogy in astronomy. So in astronomy, people also are interested in understanding how the planet moves in the space and the time. And around 400 years ago, Tycho managed to force the big data observation of the planet evolution. And then his student Kepler made analyzed the data very carefully and also proposed the three laws of planetary movement, which eventually led to the discovery of the gravitation theory of Newton. So to me, it seems like nowadays we are already able to generate the big data, but we also wanted to have some way to move beyond the description and also just annotations to get the mechanistic and predictive models or series of cell state transition. In my view, in the past few years, there are quite a few methods tries to achieve this overarching goal. So one of the most exciting approach in this direction is the ion velocity. In a very innovative work that published recently, Nemona and others found that although majority of the single cell particle tries to capture the fibrinogen sequences, they also capture around 20% of the introns in addition to the exons. And then, based on a simple physical model that describes the RNA transcription, splicing, and also degradation, they are able to derive a quantity called ion velocity, which is essentially the spliced, the time derivative of the spliced RNA. And because in order to make this, in order to analyze the data more easily, so they make a few assumptions. Firstly, they treat the splicing rate across all the gene as a universal constant, beta equals to 1. So then the ion velocity is defined as U minus gamma S. Here, U is spliced RNA. Sorry, the unspliced RNA, S is the spliced RNA, gamma is the degradation parameters. So U and S are measured. We only needed to estimate the degradation parameters. And then in their paper, they treat the cells on this spliced and unspliced phase plot. Sorry, the cells, the diagonal of this the diagonal of this spliced unspliced phase plot is cells then across and then, so it's actually to a low-dimensional space to reveal the direction of the cell movement. And this method is actually tend to be very popular and very exciting in the field. However, it still have a lot of limitations. So first of all, as you can see, it only tells you the short-term dynamics and magnitude of the cell condition. Secondly, it mostly used as a descriptive instead of a predictive tool. And also, since it assumes that transcription splicing rate is a constant, so the ion velocity is actually relative. And lastly, which I will also show later, they made this approach implicitly assume a constant transcription rate, which sometimes will lead into profound errors. So in the Wake field paper I just mentioned, we showed that using the dynamical framework and the metabolic labeling, we are actually able to overcome some of the fundamental limitations from the conventional ion velocity analysis. So basically, for this, we generate a metabolic labeling based HCRNA-seq experiment. So we grow the HSC cells in vitro for around 4 or 7 days, and then we use a phase U to treat the cell to label the nascent RNA, and then we will harvest the cell for SCNT-seq to generate a metabolic labeling argument, single cell RNA-seq. Then we also developed another tool for the dentist to quantify the new total spliced and unspliced RNA, and then we can use the spliced and unspliced RNA to perform the conventional ion velocity analysis. Similarly, we can also use the new and the total RNA to perform the total ion velocity analysis with dynamical. So interestingly, no matter what the tool we use for the spliced RNA analysis, the ion velocity flow is always problematic. You can see, based on the screen name, the cells from megakaryocytes, basophil actually moving backward to progenitor. On the other hand, if we use the total RNA based on the labeling date, we see the ion velocity flow is actually perfect. You can see the cells are deviating away from the HSC to become occupants of terminal cell heads. So we are wondering what's leading to this problem for the conventional ion velocity. So we specifically checked a few marker genes, for example, the PF4, which is a marker for the megakaryocyte lineage. We see that this gene has a sudden or rapid induction in the megakaryocyte lineage. So as I mentioned previously, the original ion velocity framework make an implicit assumption of the constant transcription rate. And simply because of this, you can see the spliced ion velocity, which is gene, is all negative. For our total ion velocity that are based on the labeling data, we can observe false positive and also negative ion velocity. And here, this figure also present additional support for this reasoning. So you can see when you for the conventional ion velocity, since they use the constant transcription assumption, the majority of the cell will below the steady state fitting name. So this name, and if a cell is below that name, the ion velocity is negative. For our total ion velocity, because the transcription alpha is modeled as this function is shown here. So this function contains the new RNA and the degradation parameter gamma, and also the labeling time t. Since this new RNA is measured, so it's different for each gene in each cell. Therefore, the transcription rate based on our framework using the labeling data it's a parameter that change for each gene in each cell. Says they automatically eliminate the constant transformation alpha, and then eventually leading to the correctness of our velocity flow. So now I talked about with the labeling data and our framework, we are able to get improved ion velocity. But of course, I'm not. This is just our first step. We would like to move beyond the ion velocity flow to obtain a function of the cell state transition. And fundamentally, our approach is based on dynamical system approaches. So under this perspective, the cell is a draw here, the S1 to S3 can be represented as like a point in this high-dimensional space. So each dimension corresponding to a particular gene. And then the evolution of this cell, which is x dot, or the velocity of the cell state, is a function of the underlying regulating network, which can be represented as fx, or a weighted field function. So in general, this network is not known. Therefore, we also don't know the weighted field function A. But we sometimes do know some of the motifs, like this self-activation and the mutual inhibition network motif. So since we don't know this network, then the question is how can we actually learn this network and obtain the weighted field function fx? We approach this problem and formulate it as a machine learning problem. As I mentioned, with the single cell and single data, we are able to measure the current cell state X. We can also using spliced or total RNA to estimate the time derivative of X, the velocity. And then the problem is how can we take these two X and data X, dt, ds over dt, to learn the weighted field function. So without going into the details, we learned, we developed a approach and we load this function, which is continuous and also differentiable, which means that once we have this function, and then you can treat this function as the ordinary differential equation. So here we gave some initial state of the sorry, this is actually a real data set for the zebrafish pigmentation. So if we give some initial state of the progenitors for this vector field function, we are able to actually predict how the cells evolved over time. So it provides us an estimation of the possible long-term prediction of the cell fate. Of course, after we learn this vector field function, we can do much more than just predicting the future cell state. Here, I'm just going to highlight a few other applications. One of the applications is it can help us to gain the functional understanding of the cell fate transitions. So specifically, in we know for the human hematopoiesis, the megakaryocytes, so this cell type shown here, tend to appear much earlier based on a lot of mouse experiment, in vitro experiments. But people actually don't know what's the underlying mechanism of this early appearance of the megakaryocytes. So then we analyze this data and we learn the vector field of the human hematopoiesis. And then from this vector field, we can automatically lump it into a hierarchical tree structure. So the HSC is located on the top, and through hierarchical progression, it become all kinds of terminal cell types. And this tree is actually perfectly match up with our known biology of human hematopoiesis. And then next, we can also take in this vector field to estimate a measure of time for each cell in the developmental process. We call this a vector field based sugar time. And then if you plot the cell development, we can see clearly. We are also able to observe the megakaryocytes. It's the first mature cell type to be appeared. And next, we specifically look at a few and also I see the other hand, and the field principle, so that we can actually calculate the iron Jacobian from this continuous differentiable vector field. So I think probably all of you already know that Jacobian can tell us essentially in this case here, if we change the expression of free one, what's the change of the velocity of the KLF1? So if it's negative, that means it's a repression. If it's positive, it means it's activation. So in this case, we show the Jacobian from free one to KLF1 is negative. So it indicates a repression. And similarly, we also did the Jacobian analysis for other interactions. We found that the free one also has self-activation, and similarly, KLF1 has a repression to the free one. So collectively, this indicates that the mass regulated free one appears much earlier in the megakaryocyte, in the progenitor cell state, and they also maintain its self-expression through self-activation we are also repressing the expression of KLF1. Therefore, it will prohibit the maturation of aerosols and promote the early appearance of megakaryocytes. So, yeah, so I just mentioned we are able to use this continuous weighted field to gain some functional understanding of the human hematopoiesis. Here, I'm going to also tell you we are able to leverage this function to make some non-trivial predictions. Specifically, we are interested in predicting the cell-fate transitions. It's maybe you already know that nowadays we are able to reprogram one cell state to another, and because of this, it becomes really fascinating question in developmental biology on how can we essentially convert any cell type to another any other cell type. So in the context of hematopoiesis, we know that we have some like the differentiation from HSC to each of the terminal cell type, and we also know the corresponding transcription factor for each of the transition. Similarly, we also know, because I'm showing here, the megakaryocytes can be reprogrammed back to HSC. Similarly, we also have some transdifferentiation between the terminal cell type. There are a lot of unknown conditions shown here. So the goal is that how can we actually develop a general approach that is able to predict the cell-fate transition, which not only matches up with our known biology, but also potentially predict the accurate transitions of other cases. So the method I was using is actually borrowed from a principled approach that was used in physics for many years. This is the least action path method. So essentially what the least action path method is that it will perform an optimization among for the optimization to find the optimum path from one cell state here 0 to another cell state among all possible passes. So those are all possible passes, but the method will eventually find the path, maybe this one shown in the red, which has at least action. And with this, then we are also able to prioritize or maybe calculate the contribution of which transcription factors to this path, and then we can give a priority score for the.
I think in order to have time, is probably finished.
Sure, just 1 minute. Yeah, so we then apply this to predict the development trajectory, and also we reveal again the megakaryocytes appear much earlier, only taking like 31 hours to come in. And also our prediction is actually really good with the AUC model called at the fair. Okay, so lastly, I also wanted to show that we are able to make some in silico perturbations. So the idea is that since now we have a weighted field function, we can introduce some perturbation to the system, essentially propagate this perturbation along the network, and then we can observe the genetic effects as well as the outcome of the cell-fate transition. And here I show that if you lock out this GALA1, you can see the cells actually moving away from the aerosol MEP cell to become GMP cells. And then if you lock out the other cell, other genes, SPA1, you observe a reverse transition. And lastly, if you lock out both genes, the cell actually trapped in the middle. All of this prediction match up with previous experiments. All right. So I think I hope I convinced you. We learned we developed the methods that they learn, they go through the velocity, which overcome some limitation from space time way analysis. We learn the continuous field function, which can gain functional insights by perform Jacobian and other differential geometry, and also we can do a lot of predictions. Right, that's all.
So at the very beginning, you showed us this manifold hypothesis. It seems to me, maybe I didn't understand parts of the talks, that the perturbative RNA-seq gives you like basically the tangent space at some isolated points of this manifold. So, like first of all, do these tangent spaces all have the same dimension so that they're all part of a common manifold? And then secondly, in order to obtain this whole manifold, like wouldn't you need to extract data when moving along the paths and not just at the source and target?
So to some extent, was what the second half of the talk was the dynamics of how you go from one state to another and how you can predict that. And in a way, yeah, so I think the notion of perturbations was really how you can better understand as you change the expression of genes, how you change the final state. But it's purely an equilibrium. And I showed you examples of how you can get information from that type of equilibrium response. But for sure, everything is dynamic. And that's why doing these single cell experiments where you do an initial impulse, like immediately turn on the gene or add a drug, or start differentiation, and then do the metabolic labeling so you can see not only where the transcriptional state is changing, but the derivative. And you actually can even do an acceleration if you do 2 metabolic labelings, lets you get insight into the dynamics of the process. So it was a lot I realized to digest, but they're really meant to be how equilibrium perturbations and the dynamic measure of perturbations can be measured by single cell experiments in a very rich, data-rich way. And that a lot of the challenge now is not the experiment, but the ability to extract information from these data.
So can you actually reconstruct the whole manifold?
I think parts of it you can, like the transcriptional aspects of it, but as you alluded to, that's not the only thing. That there's clearly, if you were looking at genes that were impacting the motors, that they may have very small transcriptional changes, but have much bigger changes in the sort of overall three-dimensional structure and organization of the cell. Or things like phase transition, liquid phase transitions that you see that are probably going to be fairly invisible to transcription.
Maybe a silly question as well. So there you show this single perturbations on single gene at a time. Would be valid to simulate the breed something that.
Yeah. So
what is this missing?
For sure, combinations that where you get neomorphic properties, there's a great deal of interest in. And of course, it's combinatorial expression that lets you have such a diversity of cell types with a small number, 10,000 genes or 20,000 different proteins, 10,000 expressed in every cell. Yet you can get this huge diversity of cell types, and that's purely from combinatorics. So looking for nonlinear effects is a lot of what Tom Norman did when he was a postdoc and continues to do. But again, I think being able to have a rich readout made it much more interpretable, because if you're just measuring growth, once you kill the cell, it's dead, and there's everything. Death is epistatic to everything, so it sort of obscures everything. But you can actually die in many different ways. You can differentiate it, or you can go into apoptosis or paraptosis, and each of those will have a different signature. And you can then see an example of this that actually was from this there's the cell line that we worked with was a leukemia cell line, and there's a therapy that by inhibiting a protein KDM1A causes the cells to differentiate to granulocytes, and the idea is that once it differentiates, it will stop dividing. But we were able to then find another phosphatase that, when inhibited in conjunction with KDM1A, is much more effective at causing them to differentiate. And so that would suggest that the inhibitors, and there are inhibitors, as phosphatase, would be a nice combination therapy. I'll do actually maybe the.
I was wondering if specifically on the RNA model, if that's something that's possible and like cancer tumor cells, and if so, if you could provide that interesting understanding of potential therapeutic
So the example was, it's more natural for differentiation. The examples was hematopoietic stem cells, which then can make erythrocytes, megakaryocytes, granulocytes, etc. Or differentiation, but in principle, you could apply it right to a therapeutic treatment.
Yeah. So our approach is, as you see, we rely on the velocity, right? So it's in general, it's only probably ideally only for the short dynamic process. For cancer, which in general takes maybe months, even years. If you don't have enough sampling and non this process, this approach will be limited. Yeah, that's just answer your question. Okay. Actually, I also wanted to respond to the first question just a little bit. So in my view, my approach tries to learn work the field function in the normal process. When you're talking about the perturbation, so you also remember, we also my approach is also able to perform the in silico perturbation. So effectively, you can also do you don't even need to do the perturb-seq to perform to know the perturbation effect. On the other hand, it's also possible to when you train the machine learning model, you can also take this perturbation as an input. So potentially, we also improve the learning accuracy, and you may be able to generate two more perturbations. Yeah.
So talked about understanding like differentiation in human cells and things like that. I'm wondering if this kind of dynamical model has been already achieved for simpler things like a bacterial cell, like you predict the dynamics of the bacterial cell.
Yeah, I think if you, in our introduction of that paper we're talking about, we actually also mentioned this early works on actually trying to build like a whole cell model for bacteria, including E. coli, and I think there is another bacteria. I think maybe it's possible to we using our approach. It's possible also build the dynamic model for just bacteria. But the key here is the key thing here is that we need to measure the velocity. So for bacteria in general, they don't have introns. So I mean, if you can also do metabolic labeling, maybe it's possible. But yeah, that requires
it's a bit of an it's a bit of an irony that a lot of this stuff was developed in simple genetic systems like bacteria and yeast, and now those for a number of technical reasons, like having a cell wall and them being very small, these types of single cell approaches are much harder. And with what we didn't have until 2013 or so was a way of changing the expression of the different genes. So it's really been, mammalian systems are increasingly not only the ones we care about because we're people, but also the ones that are experimentally most amenable.
So earlier in the slide, second part of the presentation showed this 85 AUC predicting the final solution. So would you speak to compare like how does that compare to a more naive baseline model? I'm trying to understand the like the modeling power does that keep you approach? If it doesn't make more naive, we say like predicting for state, final position, or like 100%, because I see that there's much benefits in being viewed as such, but how does that the raw performance in the past compare to other modeling methodologies? How does it compare to seeing benefit? Just that's a prior.
So what other models are you talking about?
I think just like naive, what.
Yeah, so you showed one particular model methodology to make this work.
Yeah.
So you could actually model as a let's say a field you say predicting the next step, like a current one that will predict the next step. Or you could have done like some even ignore future positions which predicts from whatever it is state the model. Like you could have this performance for it. I'm wondering how to speak to the technical advantages of the approach.
So I think we did some benchmark to benchmark the performance of our approach. Basically, we simulate the dynamical system. We know the exact function of the system. And then we use this approach to learn to reconstruct that system. But the goal for us is we wanted to actually able to gain this predictive insights. So we wanted to get the calculate the acceleration, curvature, Jacobian. I think for most of the approach, since I don't think it's differentiable. You can't calculate the differential, right? You can't calculate the acceleration. You can't calculate the second derivative. So in that sense, our approach is unique. But we do compare the different method to reconstruct this vector field. We compare the kernel methods based on HK Hilbert space. Also, we also we also recently have a new method that relies on deep learning. What we show is that in general the kernel method works well, but for the prediction on the boundaries of the dynamic system, it tend to getting worse.
That the deep learning was doing better.
Deep learning doing better on that sense. Yeah.
Yeah. And I say one other thing, maybe just to give a perspective on this, that these labeling where you put in a metabolic label into RNA, so it can really define how old the RNA is, and to read that out in a single cell is just something that's come up in the last couple of years. In fact, the big limitation that Jajay had was he was working with experimentalists to get these data sets, and then the pandemic happened. So he basically submitted the paper on existing data sets and then got the reviews back and worked with someone in Vijay Sankaran's lab to collect the ThioU data set that really showed the power of this. So that it was a situation where these metabolic labeling, large-scale experiments, ability to measure RNA velocity accurately was getting ahead of it. There was no real analytical framework because there was no data until a year or two ago. So.
Last question, Peter.
Yeah, I just have one question about part of the methodology where you say you're trying to find like the least function.
Yeah.
Like what is the reason behind that? Like, could it be possible that you have that path takes you to this, the sulfate that you want, but it's not necessarily the most stable sulfate, and so like another path would actually realize a more stable population of cells with that sulfate? Or,
I think maybe this structure path maybe not the best approach for real biological system, but we need something that operational. I mean, and we show with our predictions on unknown sulfate transitions, it works well. So that's why we use it. But in the future, we may develop other methods based on different assumptions.
And do you evaluate how stable it is once it gets there, or does it like revert to?
In general, so I think I didn't mention this in my talk, but when we do this prediction, we are predicting between different steady states or attractors. So just a stable cell state, in general, they won't move out automatically.
Yeah, so you define states, steady states, stable states, and now you're trying to ask the question, how do you get from A to B? And it can go in principle through any other possible state, but they would require lots of differentiation, essentially climbing up a hill. So it's really trying to predict which is the path that's most likely to go, through. And the value of that is that when you predict the path, you can see what transcription factors are turning on at the highest energy point in the path. And then those are ones that you would predict that are drivers of the transition.
So thank you very much.